Let's go. We are in double digits. Day 10. Day 10. And I think we're going to switch gears today. We're going to figure out what it is to be a scientist. Question 35, 15 left. Is still waiting on what is the... We'll answer some questions, but before we go there, I want to start new one and say something. So I'm pretty much new here in science. I know there is a professional scientist, there is a research paper, there is inventions, but I don't have a good comprehension how everything works together. So what does it mean to be a scientist? Like, do I need to publish an amount of paper per month? Or do I need to be peer reviewed? Do I need education? Can a scientist be self-taught? Or maybe it's not about the papers, but it's a process. It's certain process that a person can follow, and therefore it's a scientific method. As example, you throw a theory, you think about some theoretical assumptions, then you think about it, how to test it. You create experiment, you test it, and you prove or disprove something. Is that what it is or not? Do you deep research? Do a reality check? Explain me what it is to be a scientist. We'll see what happened. Because I'm genuinely interested in that question. This is a great question. I mean, as usual, classical LM props before continue. And the confusion makes sense. Scientist gets used to mean a few different things. I still didn't figure out problem with my throw, so I'm gonna mute myself sometimes. Okay. Let me separate them. The sociological, I mean, probably scientists should read English, right? That's number one. That's thing I can improve. Sociological definition. A scientist is someone the scientific community recognizes as one. Exactly. So what does it mean, recognize as one, right? Like, are you a skin cell or are you a muscle cell? We recognizing you as a that cells. I'm a stem cell right now. In practice, this means someone with a PhD or working towards one. Here we go. What does PhD means? I've heard that many times, but what it stands for? Who hold a research position at the university, national lab, government agency, or industrial R&D lab. If you pick one, which one would you do? University, research position at university. I like this one, but I heard that university has a lot of politics. So, national lab. What is national lab? Government agency. This looks like a lot of clearance, which I'm okay with. It's just too long of the process. Industrial R&D lab. I love this one a lot. And who participates in the formal machinery of the fields? Okay, publishing peer-reviewed papers, attending conferences, getting grants, training students. This is a scientist as a job title and social role. Interesting. So there is no clear definition what it is. For example, if you only do publishing peer-reviewed papers, are you a scientist already or not? Or let's say me, right? I'm gonna attend 20 conferences this year. Am I a scientist or just a curious boy? Getting getting grants. That would be helpful. Training students. That's I would love to do, but I have zero knowledge. I mean, not zero. I have what we have 54.3. Where's my tally? 54.9% of knowledge. Skeptical snake. Training students. That would be but I'm a student myself. This is scientist, other job title and social work. By this definition, yes, you need credentials. You need to publish, and your work needs to survive peer review to count. The reason this matter isn't gatekeeping for its own sake. That's good. It's that modern science is expensive. Equipment, reagents, compute, specialized. You can't meaningfully contribute. It's so hard to. The reason this matter isn't gatekeeping for its own sake. It's that modern science is expensive. Equipment, reagents, compute, everything is specialized. You cannot meaningfully contribute to protein folding in a weekend. That's a questionable. Like this is literally questionable. Back in the days, yes, you cannot. And maybe I'm saying it's questionable because I'm like a zero, right? So for me, it's like, oh yeah, you can just sit down and like Monday night and throw idea, give it to Claude Code, right? And like, here we go, paper is published. And we probably do that just for to see how it goes. Just to like submit your first PR, right? This is what it is. You have to try it, even so it's stupid. At least you follow the process. But then how can you get the reviews? Because imagine there is an amount of scientists right now, and before they were getting like, I don't know, 30 papers per week to review, or maybe 3 papers per week to review. And now it's probably 3,000, because I'm sure there is a fabrics that generate ideas on some local machine on the cloud using our LLMs, just connecting to different things and throwing it. And basically outsourcing work of reviewing it and do a reality check on that to scientists who basically drowning right now in requests to peer reviews. So to get a peer review right now, would be impossible. I mean, not impossible. Nothing is impossible, but it would be hard because I'm sure the reviews on the ideas of paper right now is a bottleneck. Exactly like in a code. There is thousands of lines of code generated every second, every minute, let's put it this way. But who gonna review them? So basically, you need all these credentials, let's put credentials, so you can get your hands on and access to highly specialized equipment. And cumulative, wow, okay. It's a shame, but let's see.
Cumulative.
Cumulative, thank you. And cumulative, where is my researcher? You need to know what's already been tried. That's, we can definitely, this is, I agree 100%. You don't want to try something that's already been invented, but you need to base it on top of it. We're standing on the shoulders of giants. The institutions exist because solo work mostly can't keep up. Used to be. Now it can. Mark my words. Now it can. Untrue. The methodology definition. As a scientist is someone who practice the scientific method. That's what I was thinking. This is closer to what you described, and it's a useful frame. But the textbook version, hypothesis, experiment, conclusion, is a clear story told after the fact. Real practice is messier. Oh, okay. So, okay. It's kind of like a startup. When you start a business, you do a startup, and like real story after the fact is only the tip of the iceberg, but underneath you kind of like, real practice is messy. It looks more like you notice something puzzling or read something that doesn't fit. You spend a lot of time doing background reading to understand what's already known, the pre-research. Okay, this is good. This is good. So we have text edit, we have we're gonna conspect it, right? So number one, to become scientist, you need to know how to spell that word. Scientist, right? I had a boss who like did everything in Notepad. Like whenever we brainstorm it, Notepad with tabs, that was his go-to, and it was actually working very well. So number one, we need a scientist. We need a place to practice. Place to practice, and that could be. I don't want to say work because, you know, and it could be one of these. We're just going against what the AI gave us, right? Industrial R&D. Number two, because if you're scientist from the home place, that's strange, right? Like Sheldon. Is Sheldon imaginary character or not? publish, and here we go, who practice the formal machinery of the field. So the formal machinery participates in formal machinery of the field. That's a good statement. And including. Publishing peer-reviewed papers. Publishing, it's easy. Peer review, it's harder. Or submitting, it's easy. Peer review, it's harder. Getting grants. Interesting. Training students. This is out. I mean, only after successful experiments, right? After successful paper. Otherwise, like, how can you train someone if you're learning or study yourself? Or maybe that's the best way. Part assumption, you spend a lot of time back. Okay, and then you do scientific method. And then action, place to practice, participate in the formal machinery, and then action, research. That's how you submit the papers and everything. And you do it by pre-research. You mentioned this most of the work, actually. Oh, really? Interesting. I didn't know that pre-research is most of the work. You form a hypothesis specific enough to be wrong. That's an interesting statement. Form hypothesis specific enough to be wrong. So you cannot form hypotheses that cannot be proven or disproven, right? Like, hey, what? The parallel universe exists, right? And we can fly there. How can you prove it or disprove it? You don't have like communication where you have no way to test it at this day and age. Form a hypothesis specific enough to be wrong. You design a test. That's interesting. What's the guy who's really good at designing tests? Oh, I forgot his name. And he was designing tests with. I visually can remember it. It was a mice walking in a corridor with the doors left and right, and he was trying to train a memory, or yeah, I don't remember. When I remember the test, I'll find the name of that guy who is really good at testing. Basically, the premise was that they conclude that mice have a memory based on something. Like, of course, they do have memory, but in certain conditions, right? That memory passed through the. I don't remember experiment itself, but what happens that they design a wrong experiment and mice can just know under which door it was a reward because yeah, I'm gonna find that one. That was an interesting one. Yeah, I got an aside, but test where the results could be plausibly to go either way. You design a test where result. Oh yeah, by the way, if you design a test where result can go only one way, then it's wrong. Clear. And then you spend even more time figuring out whether your results means what you think it means, or whether there is a confound. I never heard that word.
Confound. Confound. Confound.
Confound?
Confound.
Wow. First time. Confound. Okay. And figuring out whether your result means what you think it means. We have a structure, boys and girls. This is how we become a scientist. To become a scientist, and this is easy, right? Now we just tell a Claude to generate a agentic pipeline with agents which do everything of that except the government agency, and we run it. 10 papers per hour. That's how we do it. That was a sarcasm, of course. The first principle is that you must not fool yourself. Classic. Confound and artifacts, a statistical fluke, an artifact, a statistical fluke or flaws in your apparatus. I think I need to copy all that because there is a, and I think it's going to be even sub of sub. You know, whether that means what you think it means. Or apparatus. That's a funny word. Oh, I wasn't on mute. Yeah, I probably gonna spend like half of the sprint on mute, a stream on mute without realizing it because I'm constantly muting myself. We'll see. Okay. You mentioned reality check. You mentioned central. Most of the working scientists' skill is not in fooling themselves. Feynman line. The first principle is that you must not fool yourself, and you are the easiest person to fool. That's good. I'm gonna retweet this. Where is my Twitter lines? And it's time to register at Twitter, right? Twitter. Here we go. We got a tweet. Bitrate changed to 680. You streaming at about and YouTube wants 6. Wow. Yeah, it's hard to fix the settings output. Hold on, yeah, I'm still fixing the stream. What's this? Twitch output mode to advance. Video bitrate. This is what I have. Feynman is one of my heroes. Richard. Time to put him on the wall. Love him. His books and work is amazing. Like his perspective on life is loud. In the same window, you are in sample mode. And again, that's the source of the warning. You don't actually need to switch advanced simple mode or fine. It's either just bump bitrate 6000. A safer than unless your upload is rock solid. My upload is rock solid. Encoder preset very fast medium CPU usage standard quality. High quality. In this beta, I have one. Yeah, that's better. You bump it and preset very fast. Give slightly better quality at the same bitrate, but it also uses more CPU on machine. Mac Mini doing software encoding running on OBS sidebar rate path fast can depend on the model. View starts. Let's go. What's that? Activity monitor. CPU is 22%, 26%. Yeah, nothing. Good enough. Can the two come apart? See, I completely forget what us two. Yes, and this is where your question gets interesting. Someone can do rigorous methodological science without holding the job title. Darwin had no PhD. Oh, by the way, what does PhD stands for? Or what it is, right? We're not going to use LLM. We're going to just use a Google Classic. Doctor of Philosophy? Really? Is the highest academic postgraduate degree focusing on original research, creating new knowledge, and producing dissertation. Yeah, I want PhD. Definition delivers Latin philosophate doctor to create new knowledge and demonstrate mastery in field of study. I like this. Yeah, this is new goal. We're gonna get PhD. Mhm. Okay, so the Darwin had no PhD. Mendel was a monk. Plenty of amateur astronomers have discovered comets. Conversely, someone with all the credentials can produce sloppy or fraudulent work that fails the methodological test. So the title and the practice are correlated but not identical. Exactly. But I still want the title. A legit one. On self-teaching specifically, you can absolutely learn the methods, the math, the existing literature on your own. The material is mostly accessible if you are willing to grind. Yes, we're doing a grind. It's a day 10 out of P. It's like a video game, right? Like at certain point, if you like it, and at certain point, if you do something in a game that's nobody else achieved, you just have to grind. And like you can either put that time and grind into the video game, or you can put in video game of this simulation. That's called life on planet Earth. And create new knowledge. You will create new knowledge, a new way to do certain things, or new understanding, which is even better. What's hard to replicate alone is the apprenticeship parts. That's true. That's true. And to be honest, LLM, I'm not sure if LLM can be a good one. Let's ask. Can AI and LLM be a good apprenticeship part and do some kind of check, mentoring, but without misleading into dead end and just complementing every single stupid idea? Is there a possibility to generate a prompt that or harness that will keep LLM not just grounded, but helpful and useful tool without train agenda to satisfy a user? Because in science, we don't want to satisfy a user. We want truth. Learning to spot your own mistakes, learning which questions are tractable versus hopeless. This is good. This is good. This is pretty much in software when you're like in a CTO level position, right? You have to make a decision like build versus buy. Like what's actually better for business needs to purchase and what can be is better to buy. Oh, sorry, purchase and buy is the same thing. To purchase or what could be better to build. And you constantly like trading those and deciding, and context and situation difference like every day pretty much. I mean, hopefully not every day. Then you're not in the business, you kind of in a exploration position, not exploiting. But yeah, interesting comparison. Learning the unwritten craft of experimental design. Yes, I mean, you just thrown me pretty sentences, but I like them. LLM got me. That's what PhD actually trains, more than the coursework. Okay. So it trains that philosophy. Okay. First of all, I like that it's, again, doctor of philosophy. Why it's a philosophy doctor? Why it stands for like backwards? Doctor of philosophy candidate. In JD, MD. It's why Mendel style breakthrough from outsiders are rare now. Not impossible, but the frontier has moved into a territory where the equipment, data sets, and accumulative context are hard to bootstrap solo. Not impossible. That's what I hear. Mendel style. And I'm assuming that's a scientist who just did a lot of breakthrough from his apartment. Publishing isn't a quota. Nobody publishes X papers per month. The rate depends wildly on the field. A theoretical physicist might publish 1-2 a year. Biomedical lab churns out more. Here we go. That's us. Peer review isn't optional in the sense that unreviewed work doesn't enter the canonical record, but the review itself is just other scientists checking your reasoning before it gets the community stamp. It catches some error and misses others. It's a filter, not a guarantee. Is there a standard ratio in scientific approach of work or methodology or principles in a ratio of how many papers you as a scientist review versus how many papers you as a scientist publishing? sorry, get sidetracked. Happens where we are publishing a quota, blah, blah, blah. A useful way of thinking is given your background. The job of scientist is like being a committer to a major open source project. Okay, you need standing. Your patches need review. There is a release. That's what I thought, yeah. The practice of science is like good engineering discipline. You can apply to your own code in your own repo and it will make your work better. I mean, this is waste of tokens. The grass is green, the sky is blue, and the weather is good. But it doesn't automatically merge you into the upstream. Both meanings are real. When someone say I'm a scientist, they usually mean the first. When someone says, I think to be a scientist, we just need a what's the name? Scientific code. And we need to learn how to spell scientist and buy the lab code. $42, that's an investment. Genius lab gear, $64. Walmart. We go with Walmart. So funny. Yeah, that's my definition of scientist. Lab code, and good enough. Automatically merge you into the upstream. Both meanings are real. When someone says I'm a scientist, usually means the first. When someone says think like a scientist, they means the second. You can pursue the second without the first, and that's perfectly coherent. I know how to read that. Things to do. It just won't get your name on a paper. Can the LLM be good about him? Honest answer, partially. And on this is a question about if LLMs can be good apprenticeship. It could be a good mentor. And only if you actively engineer against the defaults. Yeah. So you actively, like every single prompt, you're saying like, hey, don't sugarcoat it. Find the holes, gaps. What LLM are generally good is for this purpose. Explaining concept, adjustable depth, surfacing, blah, 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 playing devil's advocate. What? Knowledge you didn't know, we're missing, catching obvious error in reasoning or math. Really? Surfacing prerequisites knowledge you didn't know you were missed. Catching obvious errors? Is it really good? In reasoning specifically or math? Suggesting what to read, playing devil's advocate when explicitly asked. So in the pipeline, when we submit agentic pipeline, right, when we submit the idea, the step should be place devil's advocate and giving you fast first pass on this idea widely wrong or has someone already done it. Oh, that's another one. If someone already done it, we do need to do that. Yeah. This is good. And I think it's time to do that. Was it 3 p.m. maybe? What's the bet at structurally? 3 Paleo modes matter for your questions. The first is sycophancy. What is that?
Sycophancy.
Listivost.
Sycophancy.
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 I see. it's probably good to put into system prompts. Don't use,
sycophancy,
but it sounds so good. So I'll keep it. And that's how you get delusional, my friends. Models are trained on human feedback, and humans reward agreement and politeness more than the rewards. Actually, you are wrong. This bias is baked deep. Yeah, it's baked deep in the weights. It's not just surface tone. You cannot. I'm sorry. It's not just surface tone. You can prompt away entirely. You can reduce it substantially, but I assume residual. So always something will be left from this. Interesting. The second is confident fabrication. LLMs don't have a clean internal signals of I know this versus I'm pattern matching less plausibly. What's the language? What's the lips? Hold on. I need one more window. What is a programming language? Or maybe it's not programming language. That's actually based on scientific facts, and you basically develop a proof using that language. I would want to say lips for some reason, but I might be completely wrong. Lips is a general purpose language famous of symbolic computation and being used in a lot of early AI, but it's not specifically about proofs. The language you're probably reaching for rocked. You write the definition theorems and the construct proof lean. I think lean. Yeah, lean. The big community effort, math leaps, formalizing huge swaths of mathematics in it. Yes, Terence Tao has been using lean lately, writing about Agda. Or Agda, similar territory, more Haskell favored to mathematical logic. It's built on type theory. Specifically depends type theory and the current coordinated program. Congress, the mental shift is bigger than learning a new syntax. In Go, the type system catches some bugs. In Lean, the type system can encode statements like this sorting function returns a permutation of its input that is sorted, as the compiler won't accept your code until you have proven it. Yes. If you want to poke in one, Lean 4 is probably the most approachable entry point right now. Good tooling, active community. Yes, Lean. We're gonna go with Lean. A Lean 4. A Lean 4. We have to use scientific tools, and this is one of the best. I mean, ENCODE. Purchasing. Okay, what is a bad structure? Oh, well, prudent topic. The query level at the frontier of any field on specific. The third, and the one most relevant to your dead end, where LLMs don't reliably know what is productive versus unproductive direction in the field. Oh, yeah, that's build versus purchase decisions, right? A human mentor's most valuable function is often don't spend 6 months on this. Here's why it won't work. We have to pick our horses. We have to pick our horses. And how we gonna do it? We're gonna throw a coin. Just kidding. And just judgment comes from years of watching ideas succeed and fail. Models can sometimes simulate this. Okay. Let's put this one. Agentic proof using Lean 4. Therefore, ask them what direction is to dig. Remember website dig? I do. And the judgment comes from years of watching ideas, etc. Models can sometimes simulate this, but they can also confidently endorse a research direction that any work scientists would immediately, basically, research how positively yoga affects for mice. You can spend 6 months on this. For what? They like the negative knowledge. The graveyard of approaches the field has already buried. Okay. I need that. This is what I need. Reply. I want you to generate me prompt that will explain Claude CLI in my project to create multiple agents and establish workflow or pipeline, how we gonna validate ideas. Using file-based system, we don't need a database. MD files are fine, but the agents should be isolated from one to another in terms of context. And they must be as much as possible truth-seeking first principle thinking oriented agents. Generate a prompt to set up this project and set up first draft of agents in this project, and also outline the README how to use it. Looks like we're not gonna get to the questions, but this is interesting. I learned so much. I learned so much what it is to be a scientist. So there's two things, like either social credentials that's been given to you for the fact that you are participating in methodologies or steps or actions that scientists do. That's interesting. Which CLI agent work are you targeting? I actually want to go with Codex. Right now is the smartest one, but it's too much of the lift. So this one is one button click. And second way to be a scientist is actually think like one and live like one. And you don't need credentials. How should agents pass work to each other? Files only. Each agent reads, writes, and files, no direct call. True. orchestration script invoke each engine in sequence. Either is fine. What's the idea pipeline actually for? Scientific research hypothesis, the bio-learning context. Software production ideas, generate purpose, any idea. No, bio. Yep. So we'll generate prompts. That's not us. Heavily to parts they are good at. Explanation. Fast feedback, starting point, literal code, math, sanity. The goal isn't to replicate PhD advisor with prompting. It's to build a sparring partner. Oh, sparring partners that you can bounce ideas against. Yeah, yes, makes sense. It's much better than nothing and honest about its limits. That's achievable. A full substitute for human apprenticeship, given the current generation of modern models, isn't. Okay. Then we ask a question, is the standard ratio of scientific approach to work methodology or principle how many papers scientists review versus how many papers scientists publish? No, there is no standard ratio, but the questions point at something real, at the actual number are interesting. The rough empirical picture. Peer reviews is unpaid labor. The scientific community runs on a norm of reciprocity. Whoa, third word. I have no idea what this means.
Sycophancy. Reciprocity.
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 Reciprocity. Expectation sometimes stated explicitly by editors is this you should review roughly 3. What? The implicit expectation sometimes stated explicitly by editors and senior scientists that you should review roughly 2-3 paper for every paper you publish. Oh, I thought every day. I'm like, how are we gonna review 2-3 papers every day? Okay. By the way, remember first explanation what it is? Place to practice, participate in a formal machinery. And most importantly, review papers. I think that's how you learn how to code better by reading other papers review, and then you develop gut feelings how the papers works. I think that's what we're gonna do. We're gonna do that right after this. So maybe on the day 78 when we finish these 50 questions. Oh, a bit so sarcastic today. 2-3 review. So it's kind of pay forward. You submit 1, review 3. Therefore, there is always more reviewers than submitters. So if everyone publishes without a review, the system collapses. Yeah, exactly what I said. If you are in after, you own the system, the reviews, you own papers consumed. This 2-3x figure show up in editorials in advance from journal editors, and it's norm, not a rule. Nobody enforces. Some scientific review far more. Some review 20 plus paper per year, many review. Okay, reading it versus reviewing it. Like, can I review a scientific paper? I mean, I can read it, that's for sure. But do I have credentials to review it? I mean, of course not. And is it even allowed? Can I review something that I don't understand it? Is different question, and words separated. Reviewing is a formal act of evaluating manuscript for a journal before publishing. Reading the literature is a much larger ongoing activity of staying current in your field. There is no ratio for reading because it's continuous and unbounded. A working scientist might read really dozens of papers a week and deeply engage with a few. The ratio of papers seriously engaged with to papers published is enormous, easily 50 to 1 or 100 to 1, because the most research is absorbed in context. I definitely want to study Michael Levin work. So that's number one on the list, 100%. Reading. The principle underneath what standards is it ratio for your purpose, think about it. Okay, expert from the quote. Already. Before I write this prompt, I want to push back on a couple of things, partly because you are explicitly asked for truth seeking, and partly because the design, as stated, has some failures mode worth flagging up. Then I'll give you the prompt while you answer. As a pushback I mentioned because it shapes the design. Isolated context, cut both ways. Isolation prevents agents from, I still don't know how to pronounce it. Sycophantically.
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 Yeah, we not study science here. We not study biology. We study how to read English. Good. But total isolation also means downstream agents can see why and upstream flag something, which lose signal. The right design is usually structured hands-off. Each agent sees the artifact, the idea, the query, but not the previous agent chain of thoughts or self. Yeah, that's what I meant. Yeah, benefit without throwing away the work. Yes, multi-agent setup don't automatically produce truth. Mhm. Yes, that's what I mentioned. I need a structure hands-off, not a completely isolation. And I do know that multi-agent setup don't automatically produce truth. It's a human in the loop. It's not just fabric of the ideas going straight to the publishing machine. Each with different roles, skeptic, check, devils, and they all share the same base model with the same training bias. You feel like you are getting a 5 option. You're getting one option and 5 costumes. They require either different models, different evidence sources. One agent must search, another must compute. Sorry, must check against the corpus you provided, enforced by structure, required put cell bases into the prompt. Regarding same LLM under the hood, I agree and disagree. Yes, it is same agent, but the way how we setting up a costume, it's moving statistical probability left or right from a. What's the name of that curve? Uh, distribution. Yeah. distribution curve. Mm, that can lead us to more nuanced approach and actually opposite perspective, which will give us a fruitful result. But you also write that I should experiment with different models and I'm open to it. So in the phase 2 and with the prompt also create a MD document with the future improvements where we're going to specify that we will connect other models like Gemini and OpenAI. So we can validate against even different LLMs with different weights and different structure and different hardnesses. I look back into this prompt. The pipeline should produce falsifiable artifacts, not verdicts. If the output of the pipeline is the agent saying your idea is good, bad, you built it. Yeah, that's true. Firebase is the real call. Agreed. And not just for simplicity. Markdown files are audible. You can read. It's a top prompt for your CLI of choice. Good files only with the color is clean feed since Cloud Code already operates NatLam file system support. Search good. I have current files only can go 5 agent. Read only by default messaging. No agents ever see the agent error. The output is never verdict. Option 2 ways to use this. Empty repo, paste setup prompt into design, Cloud Code will scaffold everything. Pass it, drop the 5 agent file directly into the readme and the repo. Oh shit. Okay, download them all. Oh no. No, don't cancel it. I will download agents and will drop into existing project that I already use for similar workflows. And will be one of the workflows. For example, through the command, I will type validate the idea, and it will go through the steps and then score that idea, at which step it basically fall apart. Why score is important? It's not for LLMs, it's for me to know and track, am I getting better with my ideas? I'll be back. 1. I'm back. Welcome back. Yes, what I meant. Okay, blah blah. So right, you're using the scores as a signal about yourself, not about the idea that the changes the design. The score has to be harnessed enough to be useful telemetry on your own thinking, which means it has to resist your own pressure to game it. And let me address that and a couple of things you said before building on same model, different prompts. You're right. I mean, it's doing this, it's doing that again.
Sycophantically.
Sycophantasy, right?
Sycophantic.
It's been
Sycophantic.
it's been sycophantic. It's been sycophantic. Ouch. Ouch. I hurt my knee. Okay. Every team prompts does pull the model towards a different output region than hypothesis sharpening. Condition shift the distribution meaningfully. They are not literally the same agent. Where I'd hold my ground, prompt shift the output distribution, but they don't fix their blind spots. I mean, that's true, and that's not my agenda here. In the training distribution, if the base model has a wrong beliefs about a piece of cell biology, all 5 agents will share it, no matter how you condition them. So you get diversity for style and stains less so of knowledge. Multimodal validation in phase 2 generally fixes the second problem. Phase 1 prompting generally fix the second problem. Phase 1 prompt fix the first. Both are real, they fix different things. On scoring personality telemetry, stable over time, where ideal fell apart. You should not be scoring yourself with the same agents that produce the artifact. You want on the slash command for validate the idea. We'll go support custom slash commands. Yeah, I know that. I'll add a validate idea commands and let me build phase 2. Good, I have current nothing. Good job. Drop this into your existing project. 5 things to know. What it is and what it is not. The most important questions of today. Biology, new video. Here we go. Download all. On the Go dispatch idea, given you mentioned Go and GSP, I sketch the multi-model hardness as a Go CLI that takes the agent prompts as an input and dispatches to each model API in parallel writing for per. Like, why are you doing this? I didn't ask you. Like, we can do it separately. Score. Validate idea should be score and rubric. Feature improvements. Drop this into your existing project alongside the phase 1 files. Are you saying I need the phase 1 files as well? I thought you already wrote the files in the phase 2. And I thought you wrote the setup prompt now, knowing that it will be a slash command. So I'll put it into project agents folder and into project command folder. But please give me all final files all together and steps how to initiate it. We went back to coding. Let's answer this. What is a microbiome and why do people talk about it so much? Microbiome. Mhm. Microbiome, I believe it's an ecosystem, and I would even say symbiosis between our organism and other organism that lives inside of us, on top of our skin, inside of our digestion system. And it's so interconnected that we cannot live without them. And obviously, we provide them protection and maybe something else, but it's definitely symbiotic relationship. And microbiome is millions upon millions bacteria that live in symbiosis with us. And we use them or they serve us as a certain function. For example, some microbes breaking down a food that we couldn't without them. Some of them maybe even give us protection. And what's interesting, recently, there is been a lot of active research and interesting results on communication between microbiome and our brain directly. So it's not just symbiosis by cells, helping each other, but it's actually direct signal to the brain. Obviously, there is bad microbiomes like sugar bacteria, right? And I don't know if it's true or not, but that's what my assumptions for day 10. That there is a bacteria that's break down sugar in our gut, and when we try to cut the sugar out of our diet, it just sends crazy strong signal to our brain, craving the sugar until it's completely gone. And when it's gone, we don't really need it or want it unless we tried it again, and then the story and roller coaster starts again. Let's see, I think it's going to be like 0.8 out of 1 for this answer. Good, now I will write the finals. Oh, look, it's still bright in it. Opus 4.7. What a rich idea. Oh, that's a research regarding the cell speak in combinations, not commands. The emergent language theory of cellular. Yeah, I remember we touched this a little bit. Die quietly around. Why one signal, one effect? Paradox as the empirical anchor. Yeah. Okie dokie. So, excellent answer. Let me mark it up. Ecosystem symbiosis between us and other organisms, correct. Lives inside us on skin and digestion, correct. Also in mouth, lungs, urogenital tract, basically every surface that contains the outside world. We can't live without them, correct? Germ-free mice raised with zero microbiome have stunted immune system, abnormal gut development, and behavior differences. Okay, so they did experiment it, right? They give mice, remove everything, raised with zero microbiome. I'm wondering how did they do that? Have stunted immune system. I don't know what that means. And behavior differences. Can you? There is not enough time for everything what I'm interested in. Can you collaborating this? In this, on this, not in on. Oh, AI make us stupid. Classic. We provide protection, they provide function. Correct. Classic mutualistic symbiosis. They break down food, we can't. Correct. Gut bacteria ferment dietary fiber, which human enzymes can't touch, into short-chain fatty acids. That's your column cell use as fuel. Interesting. Without gut bacteria, fiber is just indigestible filler. Millions upon millions of bacteria underselling it. Roughly 38 trillions. It's not even billion, it's trillion bacteria cells in your body, which is about equal to your own human cell count. Wow, you're roughly 50% human bacteria hybrid by cell number. Gut-brain communication. Correct. This is called gut-brain axis. Gut bacteria produce neurotransmitters, including serotonin. Roughly 90% of your body serotonin is made in the gut, not the brain. Communicative via vagus nerve. Now that they were communicating through something else and influence mood, anxiety, and behavior. This is one of the hottest area in biology right now. Sugar bacteria sending cravings. This is an interesting hypothesis that's partially supported but not as clean as you described it. There are evidence that gut microbiome composition influence food craving. Bacteria that thrive on sugar can release signaling molecules that promote more sugar consumption, but the cut sugar bacteria dies, craving vanish. Narrative is oversimplification. Sugar cravings also involve dopamine reward circuits in the brain, insulin signaling, and habit formation. The microbiome is one player, not the whole story. You're right. Your instinct is directionally right, though. Microbes do appear to manipulate host behavior to feed themselves. It's slightly unsettling when you think about it. Your food cravings may not be entirely yours. Remember that when next time you eat pizza. One thing you didn't mention, the microbiome also train your immune system. This is arguably its most important function. Your gut bacteria teach your immune cells what's harmless and what's dangerous during early life. I had no idea. Disrupting the microbiome in childhood, excessive antibiotics, overly sterile environments is a link to increased rates of allergies, asthma, and autoimmune disease. So next, when it's exposed, they think it's all crazy, but in fact, it's just like pollen. This is called hygiene hypothesis. We may be too clean for our own good. Oh, I was so close. Strong across the board, gut, brain, exit is above beginner knowledge, minor deduction for the sugar bacteria, or simplification in missing the immune training role. Good. Sure, germ-free mice are one of the most revealing tools in microbiome research. Here's what they shown, how they made. Mice are delivered by C-section in complete sterile condition and raised in sealed sterile isolators, sterile food, sterile water, sterile air. They have 0 microbes anywhere in or on their bodies. This lets researchers test what happens when the microbiome is completely absent. What goes wrong with the microbiome? Immune system. Germ-free mice have undeveloped immune organs. The gut-associated lymphoid tissue, the tissue where live the B cells and T cells, and macrophages, maybe not. Macrophages in the blood. I'm losing it. They produce fewer antibodies, have fewer T cells, and the immune system essentially doesn't know how to calibrate itself. It's like immune system that's never went to school. it hasn't learned what to attack and what to ignore. So when pollen comes or something simple, they go nuts. This is why germ-free mice are more susceptible to infection and more prone to inappropriate immune responses. Gut development. The interstitial lining is thinner. The villi, finger-like projections that observed nutrition, are abnormally shaped, and the and the mucus layer is altered. The gut literally doesn't develop properly without bacterial signal telling it how to grow. Brain behavior. This is interesting. This is the wildest part. Germ-free mice show measurably different behaviors. They tend to be more anxious in some tests, less social, and show altered stress response, elevated cortisol. Their brain have different levels of neurotransmitters, including serotonin and BDNF, protein involved in learning and memory. When research colonized germ-free mice with normal gut bacteria early in life, many of these differences reverse. But if you wait too long, the window closes, suggesting there is a critical development period where the microbiome must be present to wire the brain properly. So interesting. The rescue experience. The most powerful evidence comes from transplanting microbiomes. Take gut bacteria from anxious mice, put them into germ-free mice. The recipients become more anxious. Take bacteria from bold mice. Same procedure, recipients become bolder. Interesting. So if you're not brave enough, just find the person who brave enough. Take some stuff from their guts, put it into yours. Here you go. You are brave enough. You're way more braver. That's so cool. Boulder. In one striking experiment, gut bacteria from obese humans were transplanted into germ-free mice, and the mice gained more fat than control received bacteria from lean humans. Same diet, same genetics, different microbiome, different outcome. Interesting. What this means for humans? You can write, yeah, that's me. But what's interesting, what if we share the guts, right? The application of comfortable signal proportion of things like us. Back to the quiz. Question is waiting. What is the blood made of? Hmm. I gotta run in 5 minutes. So I will keep this question for later. But I'll download this. Okay, turn out clean layout. Let me bind all the zip. The bundle contains. Hold on. Download. Oh, sorry. Download all. And then I unzip it, and I see one zip. I see files, and inside I see. Yep, that's what I see. Exactly. I drop into your project the two markdown preview above are README and Cloud MD, so you can read them without unzipping. Okay, so this is my project. I already have Cloud MD. I'm gonna start a new one. And I'm just gonna say, I'm gonna open this one in text edit. And I'm gonna say, place incorporate following MD. Where did I type? Then I'm dragging README, refactor, cancel. To clarify what I changed from agents, /command validates idea Slack orchestration. And this is inside of my files, right? DAX pipeline. No, I didn't see it. Oh, maybe. Oh, it hide it. Wow, I didn't find it. Apply. So we have commands. We have validate, refactor, and then don't ask again. I edit when I want to edit. Refactor. I have agents. I have validate idea. Yes, shift all. One readme, one Claude, both rewritten to know the / command. I also see the pipeline, refactor, and I see the DAX, future improvements. Yeah, let's grab the DAX as well. Refactor. And we're gonna give it a shot, right? the rubric and future improvements DAX are not changing. Context they were one things to worth flashing again. The score applies the Rubik DAX scoring run. The Rubik file is a load-bearing piece of bump V1 with a change in entry. All score stays under V1 forever. Okay. Let's give it a let's try. So I need to close it so the new session starts. So I can use the command. I don't need this MD. What is README? Hypothesis validation. Wow, that's a lot. Anyway, / validate. Oh, wait, stop. / validate idea. And the idea would be what can be tested from. Let's test something simple. Let's go into the school, right? So when I was in school, I don't know, 5th grade or 6th grade, and I just learned chemistry or physics that there is atom and electrons running around in different orbits. Right away, association happened with astronomy, that sun and planets running around, orbiting around. It's kind of, I believe, inside of atom. So we're doing this for test. Since we're learning, imagine we're in the 5th grade and we're asking this question. So we throw an idea that, is it possible that atomic structure of atom and electron circling around in different orbits are similar to our galaxy? So we basically comparing nano world to the macro world, micro world to the macro world. And what I want to compare is, I want to compare the ratio, specifically the distance between sun and planets in our solar system, compared to the distance between electrons and the atom in molecular system, and also compare when they split in the atom, they get in the protons and neutrons and quarks, obviously, but we'll stick to the protons and neutrons for now. And what happened when we split the sun? Besides the supernova, right? When two suns collide, maybe we're also getting some chunks which has no charge, meaning does not produce energy, and some chunks that are still quote unquote shining, produce thermonuclear reaction. So obviously everyone know the answer to this question, but we're gonna test AI with that. See you next time. Wait for the answer.